Welcome to Dr. Haldar online class. Today we will be discussing on another important metabolism, that is beta oxidation of fatty acids. We all know about the fatty acids and how the metabolism occurs inside our body or biological system, especially on the beta carbon atom that we will be looking into. Before starting it, I want to tell what is about beta carbon. The carbon atom, which is primarily attached to the functional group, is known as alpha carbon atom, which is followed by the next carbon, which is known as beta carbon atom. So, usually, all the fatty acids are oxidized primarily on beta carbon atom, and the oxidation are divided into three different stages. The first step is the activation of fatty acids that occur mainly in the cytosol, followed by the transport of the activated fatty acids into the mitochondria and actual oxidation of beta acid of the activated fatty acid occurs inside the mitochondrial matrix. Brain, erythrocyte, adrenal medulla cannot utilize fatty acids uh, for their energy requirement. Now we will be looking into how the oxidation occurs using the three stages which I mentioned just now, and also it is written in the slide. The first step is the fatty acid activation. Here, when we are seeing into the structure, it is mainly an example given here of 1 to 3, 3 carbon based fatty acids. Okay, so in the presence of the ATP dependent thiokinase enzyme, this fatty acid is converted into acyladenylate. Okay, and ATP here is getting converted into PPI. Okay, and this PPI in the presence of pyrophosphatase is getting converted into 2 PPI. 2 phosphate is getting liberated out of the 3 phosphate in the ATP. ATP is made up of 3 phosphate, right? So if 2 phosphate out of the ATP is released by the enzyme pyrophosphatase, then only one ATP molecule should be remained. And what is the compound which contains one ATP molecule or one phosphate molecule? That is AMP, adenosine monophosphate. So from ATP, adenosine triphosphate, adenosine monophosphate will be produced and it will be attached with this fatty acid, and that will called as a acyl adenylate. So what will happen to this 2 phosphate molecule? That will be liberated out from the with the presence of the enzyme pyrophosphatase. Now acyl adenylate will again convert it into acyl CoA. Okay, so where what will happen, this AMP will be replaced by another coenzyme, that is CoA. You can see here. Fine. So what is the form of the acyl CoA? R CH2 CH2 C double bond O and CoA. Therefore fatty acids are activated into acyl CoA mediated or through the participation of the enzyme thiokinase. It is basically a two-step process. First step, fatty acid will be activated into acyladenylate, which will be following by the conversion of acyladenylate to the acyl CoA. Now we have acyl CoA in the cytoplasm, because these are the reactions which occurs in cytoplasm. Now let's see how this acyl CoA will be made available or will be transported into the mitochondria, because we have discussed in our previous slide that the oxidation of beta oxidation of fatty acid occurs in the mitochondria, not in the cytosol. So for doing that, we need a second stage of oxidation, that is called the transport of this activated fatty acid from cytosol to mitochondria. Here you can see the figure. It is telling all about the transport system. If we considered this as a inner mitochondrial membrane, fine, this as a inner mitochondrial membrane. So this side will be the cytosol and this side will be the mitochondrial matrix. Inner mitochondrial membrane of what? Inner mitochondrial membrane of mitochondria. So therefore definitely it will be the mitochondrial matrix, it will be the inner mitochondrial membrane, it will be the cytosol. Just think about a cell system. Okay, and we call this as a outer surface of inner mitochondrial membrane, and this part we call as a inner surface of inner mitochondrial membrane. What will we call this one as a inner surface of inner mitochondrial membrane, whereas we will be calling as this side as a just the periphery of this side is called as a inner outer surface of inner mitochondrial membrane. So in the previous stage we have seen that acetyl CoA fatty acids are activated into the acyl CoA. So the acyl CoA present in the cytosol will be converted into acyl carnitine by the presence of the enzyme carnitine acyltransferase 1. Where this enzyme can be found? This enzyme can be found, I mean this as carnitine acyltransferase 1, this enzyme can be found only at the outer surface of the inner mitochondrial membrane. And what is the role of this compound that is called carnitine? It will just take this acyl CoA from the cytosol and it will assist for the acyl CoA to come from cytosol or cytoplasm. It will help to pass through this cytoplasm into the mitochondrial matrix via a special carrier protein, a special carnitine protein. Fine. So now the activated acyl CoA is slowly is transporting towards the mitochondria, mediating through inner mitochondrial membrane via carnitine with the help of the enzyme carnitine acyltransferase 1. Now it simply it will pass through and it now the acyl carnitine is within or inside the mitochondrial matrix, where what will happen here, the another enzyme that is carnitine acyltransferase 2 will come, and what it will do, it will take the carnitine back into the cytosol for this process to continue. And rest what will happen, or rest what will be the rest product, we will just obtain as acyl CoA. Fine. So once acyl CoA this is the activated acyl CoA, right? Activated acyl CoA was first from fatty acid in cytosol. What has happened? Activated acyl CoA has formed. Now activated acyl CoA from cytosol has passed through the carnitine system, I mean passed through the mitochondrial matrix barrier, mitochondrial barrier, and it came to the mitochondrial membrane barrier, and it came to the mitochondrial matrix, and this carnitine can easily pass through this mitochondrial membrane, so it again went back to the cytosol. Now we have the activated acyl CoA inside the mitochondrial matrix. So now it is ready for the beta oxidation to occur. So these are the four steps where the beta oxidation of the acyl CoA, which has now is inside the mitochondrial matrix, will happen. What are the four steps? First step is oxidation, second is the hydration, third step is the again oxidation, I mean the second oxidation, which is finally followed by the liberation of the acyl CoA. Fine. Let us see. Now it is the activated form of acyl CoA, and here the pink color is indicating the type of the number of the carbon atom. It is the alpha carbon, therefore it is the beta carbon. Why it is alpha carbon? Because the carbon is immediately attached to the main functional group is called as alpha carbon, which is followed by the beta carbon. So all the oxidation will happen in this carbon atom. So what is the first step? First step is the oxidation, and oxidation happens mediated through the enzyme acyl CoA dehydrogenase. That means definitely some hydrogen will be taken up from this compound and it will be accepted by some coenzyme. Here what is that coenzyme? Here the coenzyme is FAD. That means acyl CoA dehydrogenase is a FAD dependent enzyme. So what it will do, this FAD or this nucleotide will just take up two hydrogen molecule from here. Here we can see it is the indicated through the pink color. So what will happen? FAD will get reduced. If FAD will reduce into FADH2, and that FADH2 utilized in the ETC cycle. Last class we have discussed about the ETC cycle. This FADH2 is the acting as the reservoir for complex 2, if we can recall, for the formation of the ATP in. So that's why it is showing as the ETC cycle and formation of ATP. So what will happen if two hydrogen molecule is liberated out from this acyl CoA? Then what will happen? Simply one double bond will form. Okay, that is specially the function of the dehydrogenase enzyme, and it will form a transenol CoA. Now this transenol CoA, what will happen? Another enzyme, enol CoA hydratase, means hydrolysis will occur. Now what will happen if hydrolysis occur in the double bond? Then what will happen? The double bond break into the single bond, and ultimately one hydroxyl group will attached here. Fine. So that is called as the beta hydroxy acyl CoA. Initially it was the acyl CoA. Now in the beta carbon, what has happened? Hydroxyl group came because of this hydrolysis reaction mediated through enol CO hydratase enzyme, which leads to from the beta hydroxyacyl CoA. What is the third step? Third step is the second type of oxidation at this carbon atomic turn, and this time it will happen through beta hydroxyacyl CoA dehydrogenase enzyme. Another dehydrogenase enzyme means definitely there will be participation of some cofactor. So what is the cofactor here? Here the cofactor is the NAD plus. So what will happen? In this NAD plus will again converted into NADH, because two hydrogen molecule will be liberated out from this compound because of the dehydrogenase enzyme. And what will happen? This NAD will convert reduce into the NADH, and this NADH will act as a electron reservoir, and which will be utilized by the complex one in the ETC cycle for the formation of three ATPs that we know already. And if the hydration, I mean this dehydration occur, then what will happen? This hydroxyl group will convert it into the ketone group. Fine. This hydroxyl will be oxidized into the ketone group. That's why it is called as a beta ketoacyl CoA. Here NAD is getting reduced to the NADS, so definitely this compound will get oxidized. That's why this CoAs, that mean alcoholic group, will get converted into the ketonic group. So it's named as a ketoacyl CoA, and beta is because of this is happening in the beta carbon atom. And what is the final step in the beta oxidation? That is the cleavage. The final reaction in beta oxidation is the liberation of two carbon fragment acetyl CoA from acyl CoA. What is happening here? In the presence of the thiolase enzyme, what will happen? There will be a breakage. Here you can see, if this is the beta ketoacyl, then breakage, where the breakage is happening? Breakage is happening somewhere here. So, and another coenzyme is coming, which is represented by the blue color. If you see the structure RCH2C double bond O, instead of CH2, CO is SCO is coming. Okay, so this is actually the two carbon less than the initial carbon. Initially it was 1 2 3 4 carbon, right? Now in this acyl group, how many carbon are there? Only these two. That's why it is written as minus 2C, means the two carbon is less than the initial carbons. Initially it was 4 carbon, now in this acyl CoA there are 2 carbons. So where the other 2 carbon went? These other 2 carbon will lead to form CH3, this part. This CH2 will become now CH3, C double bond O, and SCOA. Okay. Now this is nothing but the acetyl CoA. And this acetyl CoA, where it will go? It will go to the Krebs cycle that we have already studied. Ultimately will form the CO2. So see here, the new acyl CoA, which contains the new acyl CoA, which contains two carbon less than the original, re-enters the oxidation cycle. It will again re-enter into the oxidation cycle, and this process will continue till the fatty acid is completely oxidized. Okay, so this is all about beta oxidation of fatty acid, and hope this discussion will really helpful for the viewers. Thank you.